Hi class, and welcome to this last lecture for Chapter 2 about chemistry for Bio 101, Introduction to Anatomy and Physiology. In this section we're going to be talking about organic molecules, or the molecules of life. Now this can get really complicated and really difficult. I'm just going to hit the highlights. There are a lot of other videos on the web that talk about organic molecules, the basics. If you look up videos about the basics of chemistry for biology, then you'll learn a lot about these things. Students, I don't want to set you up for failure at the beginning here, but students often struggle with this part, just kind of sorting everything out and figuring out what goes where. So I'm just going to try to give you a little bit of an overview here. Please read the section in the book and look through the PowerPoint and see how everything is all connected together here. The most important thing to start with, though, is this idea. So the molecules of life are all organic molecules. And we know that they are organic because, as we talked about before, they have carbon and hydrogen. So those are the building blocks, those are the atoms that build all of these molecules that we're going to be talking about. Now, when we're talking about organic molecules, we have four main types. All of the organic molecules are going to be put into one of these four groups. The first one is carbohydrates, second one is lipids, then proteins, and nucleic acids. And the other nice thing, I guess, about this part is that these are the building blocks of cells, these are building blocks of life. And so we do see these all around us, and they're important parts of our diet. And that's probably where you've heard of carbohydrates and proteins before. Specifically, maybe you've heard of lipids. But if you take any type of food and look at that food, turn it over, you're going to have these nutrition facts. If you look closely at the nutrition facts, you'll see that, oh, these are all organic molecules. So we have the calories at the top, and that's really just saying how much energy you get out of these organic molecules. But if you look at the next one, you have total fat, you have saturated and trans fat, and cholesterol. Those are all what we consider lipids. So lipids are fats and oils, including cholesterol. Now sodium is not an organic molecule because it doesn't have carbon. It's just a salt. We just put that on nutrition facts because salt is important in our diet. Then we have carbohydrates, dietary fiber, sugars. Those are all carbohydrates, as they say, as the word says here. And then proteins down here at the bottom. The only one not listed is nucleic acids. Nucleic acids are what build DNA, and that's our hereditary material. And so that's not something that we have to get in our diet. DNA, we're able to build the molecules of DNA as long as we're getting all these other things in our diet. So there you go. You have lipids, you have carbohydrates, you have proteins. Now this thing doesn't, whatever this is, doesn't have any proteins and doesn't have any fats. It's just all carbs. So just kind of as you're learning these things, you can look at your nutrition facts and kind of follow along there. The next thing here, so these molecules are what we call macromolecules. They're large molecules: carbohydrates, lipids, proteins, nucleic acids. Large molecules built of smaller subunits. The words polymer and monomer are important here. So macromolecules are polymers, meaning that poly means many, so we have many subunits coming together to form polymers. The subunits are called monomers. Now each of these types of organic molecules have certain monomers that are used to create the larger polymers. So as we look at each of the types, we're going to see those monomers and polymers. Those monomers are hooked together by a type of chemical reaction called a dehydration reaction. Dehydration meaning that water is removed. So by removing water from these two monomers, it actually connects them together. The opposite of the dehydration is hydrolysis. By adding water, we break these macromolecules apart. That may sound kind of counterintuitive. By pulling out water, this picture here shows how this works. By pulling out water, we combine them together, we create a covalent bond. By adding water, we break them apart. So here's a subunit, a monomer, here's a subunit. You take an H group from one and an OH group from another, you pull that out to create a reaction which binds the two together in a covalent bond, and then opposite for hydrolysis. So dehydration, pulling out water, bonds things together. Adding water breaks things apart. And that's true of all these organic molecules. So let's start with carbohydrates. We're going to look at the monomers and the polymers and then some examples. So carbohydrates, they contain carbon, hydrogen, and oxygen, and that's it. It's in a specific ratio. And these guys, the main job here in our body is for energy. Carbohydrates have other jobs in other organisms, but these are the simple carbohydrates. We call them monosaccharides. These are the monomers of carbohydrates, the monosaccharides. There's glucose, fructose, and galactose. You've probably heard of these before. So glucose is a building block of carbohydrates. Now if we take two glucose molecules, the monomers, and we bind them together using a dehydration reaction, we get what's called a disaccharide. Saccharide means sugar, so di means two sugars, monosaccharide means one sugar. So if we take two glucose molecules and bind them together using a dehydration reaction by removing water, we end up with something called maltose. Maltose is a disaccharide. Maltose is a polymer. It's made up of two glucose molecules. And sucrose and lactose are two molecules that come from the combination of two other monosaccharides, as is shown here. And you've probably heard of all these: sucrose table sugar, lactose milk sugar, maltose grain sugar. So we can further take these molecules and make other polymers. This slide here shows how you can take two glucose molecules. So this is the chemical form of a glucose molecule. So you can see all the carbons and hydrogens in here. And here's another one. Now we can also draw it like this. This is still considered a glucose molecule. All they've done is they've taken off the carbons and the hydrogens, and they just become understood, because it just becomes complicated to write them all every time. So if you see this little hexagonal shaped molecule, then that's going to be a glucose molecule. Now by taking a hydrogen from one and a hydroxide from the other, we bond these together and we form maltose, so two glucose molecules. Okay, polysaccharides. So what if we add even more glucose molecules together? Then we get these even longer molecules, such as starch. You've probably heard of starchy foods like pastas and potatoes and things like that. So starch is just a whole bunch of glucose molecules bonded together. This is how plants store glucose. Plants need energy just like us, and they get it in the form of glucose. Glycogen is just like starch, or similar to starch, but it's storage of glucose in animals. So if you just ate a meal and you've got lots of glucose in your body, your liver can take that glucose and store it as glycogen. And then when you aren't eating anymore and you run out of glucose, then your liver can break that glycogen down, add glucose to your bloodstream to keep your sugar levels up. All right, then the last polysaccharide I want to mention is cellulose. Now cellulose is what we call fiber in our diet, because we're not able to digest fiber. We're not able to digest cellulose. If you come to this nutrition fact I showed you, here's your dietary fiber. Fiber is made of cellulose. So cellulose is found in plants, and that's how it gives plants structure. It doesn't give them energy. It gives them structure. And it's just made of glucose. It's these long chains of glucose all hooked together, but it's in a form that we don't have the capability of breaking down. Some organisms do, but we don't. Here's some examples. So here is starch. So all these little hexagons are glucose. Look how many glucose molecules are all hooked together in this starch molecule. That's what we get when we eat a potato. Here's glycogen, very similar, but even more branching than starch. But it's just a bunch of glucose. And then I don't show cellulose, but you can look up pictures of cellulose. Okay, the next group is lipids. Lipids, same thing. We have monomers and polymers. So lipids, the thing that sets lipids apart is that they are hydrophobic. They are hydrophobic. They do not dissolve in water. And they are just made up mostly of carbon and hydrogen. Long chains of carbons and hydrogens all hooked together. And that's what gives them makes them a good long-term energy storage. So that's where we store those things. Lipids have other jobs too. They're not just energy storage. It also is insulation. It also adds protection. In some organisms, it creates a water repellent as well. So there's lots of jobs of the lipids. There's also lipid hormones in our body, and then also cholesterol is a big thing as well. It's really important. Okay, there's two main types of lipids we'll talk about. There's fats and oils. Fats are generally going to come from animals, and they're solid at room temperature. And then oils are usually from plants, and they're liquid at room temperature. That's the kind of difference. Fats are solid at room temperature, oils are liquid at room temperature. Fats and oils are also called triglycerides, tri meaning three, and glycerides, meaning that we have three glycerol molecules. One glycerol molecule forming with three fatty acid molecules. That's how we get a triglyceride. So I don't have, where's my picture of a triglyceride? 1 second. Okay, here's an example of a triglyceride. These are three fatty acid chains. Notice how they're all carbons and hydrogens, majority. And then up here we have the glycerol molecule. This is a triglyceride. Within triglycerides, we have what are called saturated and unsaturated fats. And these have to do with the number of covalent bonds. So saturated fats only have single bonds, and these again are generally going to be solid at room temperature, like lard and butter. And then we have unsaturated fats which have double bonds, and these become oils, because what those double bonds do is they create a little kink. They create a little kink in the molecule that makes it so they can't pack tightly together, so they're liquid at normal room temperatures. So liquid, oils, unsaturated, solid, fat, saturated. Generally, saturated fat is considered worse for you than unsaturated fat, because saturated fat can solidify inside your arteries and vessels in your body and can cause blockages and things like that. So it can be a bad thing. Notice our dehydration hydrolysis reaction again. That glycerol molecule binds with three fatty acid molecules, and by pulling out water, we bind them together. By adding water, we break them apart. See how that reaction goes both ways. One other thing to mention here is we have what are called emulsifiers. These are molecules that are polar and nonpolar, kind of like soap. So if you have a greasy pan, if you put that grease in the water's not going to react with it. But if you put in soap, then the soap binds to the oil, and then the water binds to the soap, and you're able to wash it away. This becomes important when we try to digest fats later in the semester. Alright, another type of lipid is called a phospholipid, and we'll see this more in the next chapter, so I'll just mention it briefly here. But it looks like this. It has a hydrophilic phosphate head and only two fatty acid chains with the glycerol molecule. This becomes the molecule that builds the membranes of animals and plants. So you can see how these kind of fit into the cell membrane. Forms the membrane of all of our cells. All of our cells have this lipid membrane, phospholipid membrane. I told you how lipids can also be can also be hormones. Those hormones are called steroids, and so they have a job as well. So hormones, I just dropped something there. So steroid hormones are lipid-based, and a couple of the good examples are testosterone and estrogen, and these are sex hormones. And the interesting thing here, though, is look at how similar they are to each other. There are just slight differences in these two molecules, but they lead to very different outcomes. This is true of all molecules, all organic molecules. The shape and the structure of those molecules determine what they do, and that is they can be very subtle differences that can cause big changes. The other thing to mention is cholesterol. We hear about cholesterol a lot in diet type of talks and things like that. Cholesterol is actually an essential part of all of our cells. You can't have too much cholesterol, and you can have too much of what's called bad cholesterol. but do understand that cholesterol is a lipid and it is an important part of our existence. And it's also a precursor to the sex hormones that I just showed you just a second ago. So, there's some interesting things there. Okay, next, proteins. Now, proteins, they have lots of different functions. These are really what carry out most of the functions in our body, our proteins. We have structural proteins, such as our hair and our nails. We have proteins that are hormones. Our muscles contract using proteins. Our proteins move things around our body and within our cells. They protect us using, for example, antibodies. Antibodies are proteins. And they also form enzymes. So all enzymes are proteins, and they help us carry out all the metabolic reactions that are happening inside our body. So proteins are very, very important parts of our cells and our bodies. So what is the building block of proteins? You may have heard of these before, amino acids. Amino acids are the monomers of proteins. You take two amino acids and you combine them together using a dehydration reaction, and you can make these long chains of amino acids, and these become different proteins. An amino acid has three parts, and I'm going to show you the three parts here. It's kind of hard to see here, but we have an amine group, which is a nitrogen group. We have a carboxylic acid group, and then we have what's called an R group. So this is a whole bunch of amino acids hooked together, but each amino acid has those parts: an amino group, a carboxylic acid group, and an R group. And this R group is what makes them different. So we have 20 amino acids that form all of the proteins in our body. And the R group, all of them have these two parts. The R group is what makes them special, makes them different. If we take two amino acids and we bond them together, we form what's called a dipeptide. The reason we have this word peptide is because a peptide bond is a special name given to a covalent bond between amino acids. They call it a peptide bond. So if you take two amino acids and bind them together with a peptide bond, they call that a dipeptide. If you add even more together, we call that a polypeptide. And then from there, once we take all these amino acids and hook them together in these long strings, then they start to form into a shape. And remember I said just a minute ago that the structure or the shape of a molecule determines its function. And that's very true with proteins. So each protein has a specific shape, and that determines what it does. We're not going to get into this very deeply, but we have different levels of structure in these proteins. We have primary, which is just the sequence of amino acids all bonded together. We have secondary structure. Let me show you. So secondary structure, so we have all these amino acids hooked together in our primary structure. They will then start to interact with each other with hydrogen bonds, causing what we call secondary structure. Alpha helix, which is this loop-de-loop, or a beta-pleated sheet. So those are two types of secondary structure. We can then further fold it up into what's called tertiary. So tertiary means third. Tertiary structure. This happens because of bonding between the different R groups. So here's tertiary structure. So now we have these alpha helix, and here we have some beta-pleated. So you can find some beta-pleated sheets in here too. It kind of goes. Anyway, so these structures build on each other. So primary structure is the sequence of amino acids, and we have the secondary structure, and then from there the tertiary structure forms as these R groups are interacting with each other. Then finally we have the quaternary. Quaternary means four, the fourth level of structure, where different tertiary structures come together in subunits to form a larger protein. So an example of this would be, for example, hemoglobin. Maybe you've heard of hemoglobin. Hemoglobin is a protein in our blood that helps carry oxygen. Now hemoglobin has a primary structure, which is just the amino acid sequence of that hemoglobin. Then from there it forms a secondary structure, and then a tertiary structure, and then finally we have these different subunits that come together to form the whole hemoglobin molecule. Just something to mention here is that not all proteins have all four levels. Some proteins are just secondary structures, such as our hair. Some have tertiary structure, and then some have quaternary structures, such as hemoglobin. But to drive home the point of this idea of structure equals function, hemoglobin has a specific amino acid sequence. If you change that amino acid sequence, you can potentially change the shape of the hemoglobin, and then it will no longer do what it's supposed to do. And that happens. We have something called sickle cell anemia, or sickle cell disease, if you've ever heard of that. And that is a breakdown in the hemoglobin molecule. So it's caused by just a single amino acid change. So you've got this long string of amino acids, just one of them is different, which then means that the protein cannot form like it's supposed to, and it's no longer able to carry oxygen like it's supposed to, and in this case it can actually lead to death. So that's how important this structure is, this idea of structure equal to function. Now proteins can denature from a number of different things. So heat changes, pH changes, and you see this if you've ever hard-boiled an egg or cooked an egg. An egg is mostly protein, and initially it's clear, kind of a clear kind of protein, albumin, and then you put it into the pan, you fry it, or you boil it, and that heat causes those proteins to change and denature, changes their color and their type of shape. And then we use these proteins for metabolism. Now metabolism is all of the chemical reactions occurring in our body. Okay, all of the chemical reactions occurring in our body. And all these chemical reactions require enzymes, which are proteins. And they're catalysts, meaning that they cause these reactions to happen more quickly. So these reactions can happen without an enzyme, but what they do is they make it easier for the reaction to happen, and they happen more quickly with these enzymes. So that's an important part of our existence as well, is metabolism. The interesting thing about enzymes is that they're not used up in the process though. So for example, if you have an enzyme and you have some molecules that are going to react with that enzyme, what the enzyme does is it provides a place for this reaction to occur. So these substrates, or the molecules that are going to combine, they attach to the active site, which is on the enzyme, which causes that reaction to occur. And then afterwards, we now have a new product that came from the combination of these two substrates, and then the enzyme can then be reused again. Same thing with breaking things down. So for example, digestion, we have these digestive enzymes, and these are providing spaces where these molecules that we ingest can be broken down, and the enzymes can just continually be reused and reused. So there's two types of enzymatic reactions. There's synthesis reactions, where things are built up, and these require energy. Dehydration is an example of a synthesis reaction. Then we have degradation or decomposition reactions, where things are breaking down into smaller molecules, and hydrolysis is an example of a degradation. We also have what's called a replacement reaction, where things are so there's degradation and synthesis. You're breaking down and building up at the same time. It's a combination of the two. Okay, the last group is nucleic acids. So hopefully you guys are all keeping up here with all these different types of organic molecules. Nucleic acids, this is DNA and RNA. So nucleic acids are really big molecules. They're built of subunits or monomers called nucleotides. And there's three parts to nucleotide. Let me show you here. There's a phosphate group. There's a sugar ring. So nucleic acids have sugar. And then they have a nitrogen containing base. Now this part, the phosphate and the sugar, are the same on every nucleotide. The nitrogen base is what is different and creates the four. There are only four. Did in the video there are only four types of nucleotides, and we're going to look at those in just a minute. So phosphate group, a pentose sugar, and nitrogen containing base, forming the four possible nucleotides. When we take these nucleotides, these four nucleotides, and we add them together, we form these nucleic acids. DNA is short for deoxyribonucleic acid. And then these are the four types of nitrogenous bases, the four types of nucleotides: adenine, thymine, guanine, cytosine. We abbreviate these with A, T, G, and C. You've maybe heard of those before. And the importance of DNA is all of these nucleotides come together to form kind of a recipe, information to build you. And really what it's doing is it's building proteins, because that's what we are made of. We're mostly made of. So when you look at someone, what you're seeing is proteins. Our hair is proteins. Our skin is full of proteins. Our eyes, everything that we're looking at is proteins. And so DNA has the information to build the proteins that make us who we are. Another couple interesting things about DNA. It's called a double helix. It looks like a ladder, and these base pairs, or these nucleotides, are hooked together in certain combinations. Adenine always combines with thymine. Guanine always combines with cytosine across the DNA, and this is what I'm talking about. So here are individual nucleotides. Notice the phosphate group, the sugar ring, and the nitrogenous base. The base goes out into the middle here. So you can see how the A's and the T's hook together, the G's and the C's combine together all across. So the order of these nucleotides and how many there are what determine what this DNA is trying to code for, what it's trying to tell us. There's one other type of molecule here called RNA, and RNA is important in the process of going from DNA to protein. Okay, so ribonucleic acid, or RNA, takes information from the DNA to the things that are building the proteins inside our cells. And there's some big differences between DNA and RNA. We don't get into these a lot in this class, but the sugar in DNA is deoxyribose, in RNA it's ribose. The bases are A, G, C, and T in DNA. The RNA are the same, except you have uracil instead of thymine. DNA is double-stranded, RNA is single-stranded, and RNA doesn't show up as a double helix, as a ladder, it's just a single strand. And then their jobs, of course, are different. So that's the purpose of DNA and RNA. Okay, the last type of nucleotide I want to mention here is one that's really important. It's ATP, and this is a nucleotide that it is a nucleotide, but it's not part of DNA and RNA. It has a job of affecting things that need energy in our body. So it's a way of not necessarily storing energy, but energy that we get from the food that we eat is converted into ATP, is transferred to ATP, and then we can use that ATP to do all the things that we need to in our body. So ATP is really important, and it is this molecule here. Notice it looks like a nucleotide, because it is, but it has 3 phosphate groups instead of just 1. So go back up to that other picture. Go back up to the picture of a nucleotide. So we have the sugar, the nitrogen base, and the phosphate group. ATP, we have the sugar, the nitrogen base, but now we have 3 phosphate groups that make this. And so what happens is the energy is stored in these phosphate groups. ATP is broken down to ADP plus phosphate. So ADP, meaning adenosine diphosphate, 2 phosphate groups, and then an extra one here. That releases energy. Our body can then take energy from the food we eat and rebuild ATP, put that energy back into those bonds, and then we can use ATP again for the things we need to do. Okay, so that was a little overview of the organic molecules. Hopefully you enjoyed this. Hopefully it made sense, and I encourage you to ask questions and or look at some other videos and things that have been made about these organic molecules.